Well, thanks a lot for taking the time out this afternoon to be here. It's really exciting to tell you about some of the recent updates at Senti Biosciences. So Senti Biosciences is a company in South San Francisco. I'm the CEO and co-founder of it. And really what we're focused on is trying to improve the genetic engineering that can go into all the exciting cell and gene therapy modalities that we've heard about at this meeting and continue to see emerge from the field. Our ultimate goal is to try to create drugs that have greater controllability, greater precision, and ultimately greater efficacy for patients. So the way we view the world is, there's been a ton of amazing effort to try to develop new vehicles for delivering therapeutic activity into patients through the use of cell and gene therapies, T cells, NK cells, gene therapy vehicles, et cetera. That being said, a lot of the genetic engineering that's gone to those to date has been relatively limited in terms of the features that you can impart on these products. So I've sort of highlighted 4 key bottlenecks that we see in terms of broadening out the reach of cell and gene therapies today. One is, in the face of target heterogeneity, how do you more precisely distinguish disease versus healthy cells? How do you tell the difference? 2 is, in diseases that can evade your treatment, how do you deal with that if you can't overcome multiple disease mechanisms? 3, in many cases, we know there may be a narrow therapeutic window. And so being able to control or regulate that product after it's gone to patients is important. And number 4, many of the diseases that we're trying to go after ultimately may only occur in certain places in the body or in certain times. And so having a product that is static in the face of a dynamic disease condition can be challenging in terms of treating patients. So at Senti, what we're focused on is really trying to develop what we call gene circuits that can be inserted to any cell or gene therapy product to enable greater control over the product phenotype. So, as many of you guys are aware, certainly there are natural genetic networks inside of all of our cells. These are typically composed of protein-protein interactions, protein-DNA interactions, all sorts of interactions between different components. And in nature, evolution has shaped the evolution and structure of these gene networks to give rise to the biology around us. At Senti, we're really focused on creating artificial gene circuits, ones that can be inserted into cells and allow us to control various cellular phenotypes. I'm going to talk about things like logic gating, multi-arming, regulator dial control, as well as what we call smart sensors. But these are all features that we can essentially mix and match together to create products that have increased controllability. To enable this, we have to really develop a powerful engineering platform, one that can be used to design and optimize gene circuits for any downstream application. So really at the core at Senti is a team of synthetic biologists, biological engineers, who've established an efficient design, build, test, learn cycle. On the design side, we have internal computational machine learning expertise where we can ingest information, either public information or our own experiences from designing gene circuits over the years to guide the development of those initial constructs and architectures. Then there's a build phase where we can design and construct tens of thousands, hundreds of thousands or more gene circuits in a single shot. And then we've developed assays that allow us to then do very specific characterization at a quantitative level about the performance of each of these gene circuits. And this is important if we really want to try to maximize the activity, the specificity, and the performance criteria of all of these elements. Ultimately, we can learn from this, and whether it's our own internal projects or our partners' projects, it helps ultimately build a better knowledge database as to designing these for future applications. So we've really focused on 4 key classes of genetic circuit elements that we think solve some of those key problems I mentioned before. These include logic gating, so we can design cells that recognize multiple targets rather than just a single one, and be able to do some sort of logical decision. Should I kill this cell or should I not, based off of the presence or absence of multiple targets rather than just a single one. And once you are able to design a product that can respond to multiple signals, you can then start playing with the specificity of that product. 2 is the idea of multi-arming, being able to design a product that actually does multiple things at the same time. Number 3 is the idea of a dial, so being able to dial up or dial down and control what the cell is doing. Now, we can do that both exogenously through FDA-approved drugs. We can also design cells that can respond to endogenous signals and titrate their activity. And lastly is the idea of a smart sensor. So being able to try to build cells that actually respond to different types of biomarkers, not just an antigen on the cell surface, but can we detect soluble factors in the blood? Can we build sensors that detect transcription factors inside of the cell? So there's different classes of biosensors that we ultimately can construct. Because of time today, I don't really have the ability to go into detail into all of these different types of examples, but I did want to show you that we can actually plug and play these into a variety of therapeutic applications. So we have demonstrated that we can import these sort of genetic programs into NK cells. At Senti, we're focused on building an allogeneic NK cell pipeline. But we've also shown that these gene circuits can work in the T cell setting and the stem cell setting, as well as in gene therapy context. So ultimately, we believe that there's really broad platform opportunity. Similar to how genome editing is really that delete button or that edit button on your keyboard, really what we're trying to bring to the table is that broader programming capability. So at Senti, as I mentioned, we're focused on an internal CAR-NK platform. We think these cells are quite suitable for housing these sort of next-generation constructs. We have two lead programs that I'll briefly mention today. Senti 202 focused on AML, where we're using our logic gating constructs to go after multiple antigens rather than just a single one. That same set of CAR logic gates that we've designed can be used in other indications. So we are expanding that into other solid and liquid tumor settings. We also have a multi-arming platform where we're engineering cells that actually can secrete multiple engineered cytokines to really stimulate the NK cells themselves, but also the surrounding tumor microenvironment. And again, that has applications beyond just liver cancer, which is what Senti 301 is focused on, but as well as other solid tumors. We're also very excited that this year we kicked off two significant collaborations, one with Spark in the area of AAV gene therapy. So in that particular context, we're really focused on engineering smart sensors, essentially promoters that can be used to design AAVs that only turn on in certain cell types. So leveraging the same design process that we use for NK cells, that design, build, test, learn process, we've established a lot of expertise internally on how to customize and build highly specific, highly potent promoters, and that concept can be used in the context of gene therapy. We also kicked off a collaboration with BlueRock in the context of regenerative medicine, again, importing this type of control into the regenerative medicine cell therapy space. So we, like many others, are excited about NK cells. There are several features about NK beyond just their typical ones that you hear about in terms of being allogeneic, being able to be manufactured off the shelf, but there are actually some key biological features that make them really well suited for the type of logic gating and multi-armoring that we're building. So on the left-hand side, obviously NK cells have innate cancer-killing capabilities, but they're also outfitted naturally with multiple receptors, a battery of activating and inhibitory receptors that's constantly sampling every single cell they encounter to determine whether they should kill or not kill that cell. And essentially what we can do is leverage that endogenous capability, that ability to sense and distinguish based on multiple targets, but do that in an artificial setting. Number two, NK cells do serve to activate the rest of the immune system. So in a sense, we can also leverage that capability to secrete and produce additional payloads that we think can boost their activity. So just to dive in a little bit to tell you about the Senti 202 program, this is one of our lead constructs going after acute myeloid leukemia. We think there are really two key challenges that we want to overcome with this initial product. One is target heterogeneity. So in the context of AML, we know that the patient population, as well as the cells within those patients, can be quite distinct. There are blasts, there are leukemic stem cells, and there may not be just a single tumor antigen that you can go after that has that ability to cover the whole spectrum. So to overcome that challenge, we've built what we call the OR gate, allows us to essentially target multiple antigens simultaneously, and essentially allow us to cover a broader spectrum of tumor cells. Number two, one of the key challenges we see in AML, as well as in the treatment of other cancers, is this issue of on-target, off-tumor toxicity. So there's not a clean single antigen in AML that's not expressed anywhere else in the healthy cells. And so that may limit ultimately the therapeutic window of the product. To address that, I'm going to show you an example of what we call the NOT gate. That allows us to implement the logic, do not kill healthy cells. And that allows us to protect the healthy cells from being eliminated by the product, but allows us to have sort of good fulsome targeting of the AML cells. So essentially, this is a diagram of what the construct looks like. There are multiple components here. So one of the things we've gotten really good here at Senti is really optimizing multi-gene constructs, getting them to all work together. On the left-hand side is what we call the activating CAR, or ACAR. It basically allows us to target two antigens simultaneously, FLT3 or CD33. Engagement of either one of those target antigens will trigger killing by the NK cell. And that's important because we want to cover both the leukemic stem cell and the blast population with these multi-antigen targeting constructs. On the right-hand side, we've designed what we call the inhibitory CAR, or ICAR. This ICAR essentially serves as a don't eat me mediator in the cell. It engages a safety antigen. In this case, EMCN stands for endomucin. It's a gene that we've discovered internally through our bioinformatics platform to be highly expressed on the hematopoietic stem cell. not expressed on AML cells, and therefore you can think of it as an artificial don't eat me signal. A lot of optimization has gone into designing this ACAR-ICAR balance, where the ICAR can shut down the activity of the ACAR. So essentially what we're trying to achieve here is on the left-hand side to maximize on-target, on-tumor killing, either through engagement of the FLT3 or CD33 antigens. On the right-hand side, engaging endomucin, which may be present on healthy HSCs that express, for example, FLT3 or CD33, that serves as a don't eat me signal that protects that healthy cell population. So we've now demonstrated various components of the gene circuits to be functional, both in vivo as well as in vitro in a variety of different assays. Showing you here initially that we want to build an active FLT3 or CD33 bivalent CAR, and we've been able to do that. CAR NK cells in this particular model show significantly enhanced survival compared to unengineered NK cells as well as untreated cells, untreated mice. We also have other data that we've published in previous scientific conferences comparing this to CAR NK cells that only go after one of the two targets as well. Now, when it comes to the NOT gates, which we think is really quite exciting and has broad applicability, the NOT gate allows us to uniquely, again, protect healthy cells that express that safety antigen. In AML, we think this is a good initial use case because the key healthy cell we're trying to protect in this population is that hematopoietic stem cell, which has self-renewal capability. And so it does actually provide a fairly low technical bar for the first initial clinical POC ultimately. So ultimately, we aim to protect at least 10 to 20% of those healthy cell populations. I'm showing you here two architectures in the middle and on the right-hand side of this figure, where we've designed two different versions of that ICAR construct, and showing that basically in the presence of FLT3 alone, we trigger significant killing with the CAR NK cells. However, when that endomucin safety antigen is present, there's a significant protection in terms of cytotoxicity against those healthy cells in the range of 50 to 70%. Now, we have a library of these inhibitory CARs that allow us to be tunable, to tune up or tune down the level of protection. And again, this is one of the reasons why we're quite excited about the extensibility of this platform going forward. In the remaining time, I'll briefly mention some of the work we're doing in liver cancer. The Senti 301 is really focused on overcoming the solid tumor microenvironment and arming cells with additional payloads that we think can stimulate the NK cells themselves, but as well as the surrounding tumor microenvironment. There's a couple technologies that we've implemented into this product that I'll go over in the next slide, which really focus on this concept. Specifically, what we're going after in this case is going after a single target, GPC3, which has been tested before in the clinic in a variety of different settings. We think it is one of those examples of a target that's fairly clean, which is why we're using that in this situation. We're also combining that with two additional sets of payloads. We've designed a novel version of IL-15, which we called CR-IL-15. One of the unique things about this IL-15 is it has both autocrine and paracrine signaling activity. So that allows us to stimulate not just the NK cells themselves, but also other cells in the tumor microenvironment, which can respond well to IL-15. Second component here that we're building is an IL-12 secretor. We know that IL-12 is a very potent immune stimulator. However, IL-12 traditionally has had some significant therapeutic window issues. So we've put this under control of a gene circuit where we can actually regulate the activity of the IL-12 in response to a small molecule drug that's FDA approved. We have published data on this, but just briefly highlighting on the left-hand side, we have an active GPC3 CAR, which is quite exciting. In the middle, that CR-IL-15 construct, again, has both expression on the cell surface as well as very high-level secretion. So that allows us to have, again, both that autocrine and paracrine signaling activity. And the regulated IL-12 arming, we've been able to achieve over several orders of magnitude in terms of on-off ratio, which is, again, quite exciting in terms of the regulation and tightness that we can give to this particular product. Here at Senti, we are building an internal process development and manufacturing team, allowing us to take healthy blood CAR NK cells, engineer them with the CAR, expand them, and freeze them. There's a lot of expertise that's gone into actually doing this because the constructs that we're building are complex and multi-gene in nature. We recently signed a GMP facility in Alameda that we're building up to help pipeline these programs through into the clinic. I'm really excited to essentially be working on this with a great team of over 80 people in South San Francisco, including other key team members who are here. Kurt Herbert's our Chief Operating Officer and heading up business development. Philip Lee, our Chief Technical Officer, our co-founder and leading up all the manufacturing efforts. Gary Lee, a longtime executive in the gene therapy and genome editing space, our CSO, and Deb Nobleman, our CFO. So finally, just want to leave you with, really our belief that, gene circuits are going to be the core of many future products, ones that we want to try to use to enable sort of more controllability, targetability, and efficacy in these systems. At Senti, we're building a platform of CAR-NK programs that we're quite excited about, targeting the clinic in 2023. We think there's ample opportunities here for partnering. So for those of you who have modalities where you think there's a significant benefit for upgrading the genetics inside, Senti has a platform that is well suited to that sort of collaboration. So with that, thank you very much, and happy to discuss further afterwards. Thank you for the time.